Good morning, everyone, and welcome to this morning's session of the Dairy Research Conference, focusing on efficiency, nutrition and fertility. I am Siwan Howerton, Senior Scientist at AHDB, and I'm delighted to be your chair for this session. Just a little bit of housekeeping before we kick off. You will all stay muted throughout the webinar. There will be a formal Q&A session at the end of the presentation, so feel free to ask questions as we go through the speakers. You can do this as explained on the screen by opening the question panel, which should show on the right hand of your screen. You can also use this to raise any technical issues that you're experiencing during the webinar. A reminder that the webinar will be recorded and it will be later available on the AHDB YouTube channel and the AHDB website if you wish to revisit the content. We are due to finish approximately 12:40 this afternoon, and at the end you will be promoted to fill in a feedback survey. Please do take the time to fill this survey so we can improve our webinars going forward. So without further delay, I'm delighted to introduce our first speaker this morning, Dr. James McCaughern from Harper Adams University. And over to you, James.
Thank you, Chair. Sorry, I'm just trying to get the screen as we normally. There, I think we're off. Nice complicated bit of technology. Okie dokie. So today we are on I think we've got there. Copper, can you have too much of a good thing? And whenever we speak about this topic and essentially that question, I'm sort of reminded of someone I used to work for who used to say that with every party there comes a hangover. So copper is an important trace element in dairy cow diets, and it has implications for dairy cow health, dairy cow performance, and dairy cow welfare. The element itself primarily functions as an enzyme cofactor in over 300 proteins, as exemplified by cytochrome C oxidase, and a terminal electron acceptor in the electron transport chain. But the basics are that this element, in essence, is associated with everything that we acknowledge to be important in terms of performance in dairy cow production systems, okay? Whether that be milk yield, whether that be body condition score, dry matter intake, fertility, and indeed immunity. But I think an important thing to remember is that in spite of the slides that I'm about to present, in spite of the message that I'm about to try and put across and the findings that we found, is that it's important to remember that copper really is the most common trace, still is the most common trace element deficiency in ruminant diets. And the reason for that is not just down to copper itself, but down to two other elements, that being, excuse me, molybdenum and sulfur. These combine within the rumen sequentially to form mono, di, and tri, and tetra thiamolybdates sequentially. Basically, what thiamolybdates do is that the complex copper, okay, we've got our little padlock here, preventing its absorption within the animal. Now, what that means is that as molybdenum and sulfur concentrations increase within our feedstuff, the quantity of thiamolybdates within that feedstuff also increases, and the quantity of copper available to our animal decreases. Now, in instances where we get very high molybdenum and sulfur concentrations and or decreased copper concentrations, we can get occurrences of copper deficiency, characterized by changes in coat color, but we have some vastly increased economic consequences associated with reductions in growth rates, milk yields, dry matter intakes, changes in body condition score, and ultimately at the excess of the condition, death results. Now, what is the pathway of copper within the animal? Basically, we have free dietary copper. Okay, so that's copper that's not bound to thiamidolates. That's absorbed into the bloodstream, transported, bound to serum albumin to the liver. Okay, so the liver is really the storage organ and the organ of copper homeostasis within that animal. What that means is that if we have a dietary oversupply of copper, the liver will gradually increase its holding capacity of the element. If the diet is insufficient in terms of copper provision to support growth, reproduction, etc., the liver will actually secrete copper into the bloodstream in order to maintain copper homeostasis and in order to maintain bodily function. If we get excessive concentrations of copper within the liver has the ability to excrete that copper back into the small intestine via the bile. So when I think about it in my head, and really it's this analogy, and it probably takes me back to the way I think. I kind of think of the liver sort of as like a diesel tank, sort of like your indicator going from red, empty, all the way up to full and then above. Okay. So in terms of bovines, sheep and cattle are slightly different. But in terms of our bovines, if we look at our orange region along the bottom, at liver copper concentrations of 19 milligrams per kilogram dry matter or less, we hypothesize that that animal is at risk of signs of clinical copper deficiency. And you can see down here, we have our characteristic changes in coat color, okay, but that can progress through to consequences of great economic importance relative to a slight change in coat color. As we get increased liver copper concentrations, we get into the sort of the green region, the happy area. You can see our liver there, nice and healthy, gritty texture, very pinky, very purpley. But as well as copper deficiency, we can have instances of copper toxicity. And although sheep are more predisposed to copper toxicity, in bovines, we think that risk of copper toxicity occurs somewhere around this 508 milligrams per kilogram dry matter. And whenever an animal enters a state of oxidative stress within this region, we can get a mass hepatic necrosis and eventual death of that animal in a very short period of time. Now, one of the things that we should acknowledge at this point in time is that bovines actually have quite a high resilience to copper toxicity, and indeed most dairy breeds, probably with the exception of the Jersey breed, have very high tolerances for copper toxicity. So it is quite possible that you would have a large number of animals in a herd within this toxic region, but very little demonstration of clinical toxicity in that instance. So the next component of our story is where do we sit within the dairy sector? What's our, excuse me, what's our copper status of the industry as a whole? What does it actually look like? And from this previous AHDB funded research where we surveyed 50 farms to look at the copper concentration with early lactation diets, okay? And here we've got these 50 farms, if I just circle them here on the right. And what our blue line is down here, that's really our expected requirements of that animal. That's calculated according to their live weight, according to their milk yield, and you'd also have in there predicted intake, okay? So according to the performance of these animals across these 50 farms, in order to meet what they need to produce, they only required really 11 to 15 milligrams per kilogram dry matter, okay, at standardized sulfur and molybdenum concentrations. But what we found was actually that copper was vastly oversupplied in general. Okay, so 40 of the 50 farms were feeding above the industry maximum guideline at that point in time of 20 milligrams per kilogram dry matter. And indeed, 6 of the 50 farms were actually feeding above the then EU limit of 40 milligrams per kilogram dry matter. Now, it's important to remember that this limit has been reduced to 34 milligrams per kilogram dry matter in 2016. So our picture within the dairy sector is really, especially within lactating cow diets, is this vast oversupply of copper. Now, as we previously said, dairy animals have quite a high resilience. So the main question that we have here is that we're oversupplying copper. The animals we're supplying them to have a high level of resilience. And it is that question that in the absence of clinical toxicity, are we actually harming the performance of those animals? And this is reflected in cow data where abattoir liver that has been monitored for liver copper concentrations contains about 40% of dairy called cow liver contains either toxic or high liver copper concentrations. But despite this, we are still feeding too much. There may be a whole variety of reasons for this. If we look at recent surveys, farmers, quite simply, the weight of evidence is that they do not want to deal with this issue themselves. They want the feed provider to essentially deal with it, or someone of that nature. But there's a general perception, and it's because we have so much resilience within our animals, that excess feeding is okay, and to a certain extent it may be, although some of the evidence that we'll present might be to the contrary. We're unsure about the effects of antagonists. They vary. Sulfur and molybdenum concentrations will vary according to diet type, according to the feedstuffs that we put in there, even within different areas of our own farm. And the other question is, we simply don't know how much is fed on farm. We have a whole variety of sources, whether it be water, whether it be our forages, whether we be supplying boluses, whether we be supplying mineral licks. And quite simply, there's no one keeping track of all those sources that are supplying quantities of copper on farm or adding them together. So this led us to this general hypothesis. We have animals that are being oversupplied. Those animals genetically are likely to be highly resilient to toxicity. But the question is, prior to the demonstration of the clinical disease of toxicity, are we actually harming these animals? And this was really the concept of these long-term studies that we're about to present in the next few minutes. So we took, we took 80 heifers, paired them at 4 months of age, and we allocated them to one of two dietary treatments. Okay, and you'll notice these numbers from our previous slide. We selected a control of approximately 15 milligrams per kilogram dry matter. That was designed to be slightly in excess of requirements. Okay, so slightly on the positive side of requirements. And then we went for a high, okay, within those two treatments, which was supplied with 30 milligrams of copper per kilogram dry matter. That contained the controls 15 plus an extra 15 to give them 30, supplied by the means of a copper oxide bolus administered every 6 months. The hypothesis being here is that the 15 is close to requirements, but that the 30 is comparable to what we typically see as a mean being administered on farms according to our previous surveys. This was a very long-term study. We sampled them between 4 months of age and right the way up to approximately 28 months of age. Whole variety of measures to include live weight gain, per body condition score, intakes, milk yield, immune challenges, fertility. To measure their actual physical copper status, we took liver biopsies at 7 months of age, 13.5 months of age, pre-calving, and then again at week 14 of lactation, with fertility being measured during the rearing phase and subsequently again during early lactation. So the question is, what did we actually find? So if we look at this table, this gives us our live weights, body condition scores, and changes in our wither height. Wither height simply defined as the height up to the shoulders, across the top of the shoulder blades in this instance. And you can see we have our control as designated by our C, our high as designated by our H, and potential statistical difference between the two. The first thing to point out is that we have no difference in initial live weight or no difference in final live weight, but we did get this slight difference in live weight gain. We're over the duration of the study. Okay, so this table only takes us up to pre-calving, so that stopped at about 22 months of age before they were biopsied in the lead into calving at 24 months of age. You can see that there was this 0.04 kilograms per day increase for our animals on that high dietary copper concentration. Okay, so you think back to copper as a growth promoter in pigs, we do see a slight increase in live weight gain. Now, is that a numerically significant number, 0.04 kilograms per day? Probably not, but we do see that increase in performance as a result. Moving on to condition score, you can see obviously no difference in initial condition score, but we do see a greater final condition score as well in those animals that were on this high dietary copper concentration, and that resulted in a greater increase in condition score across the duration of the study. Okay, so We've got two here, slight increase in growth rates. We also have these animals with a higher body condition score. So those animals that were receiving more copper were both growing faster and actually looked better. Okay, actually looked better to the eye. No difference in wither height. So moving on to our measures of fertility. If we start with our first few measures of fertility, we've got our first observed estrus and days and our live weight of that first observed estrus and days. You can see that our highs not only came into first observed estrus at a younger age, they also came into our first observed estrus at a lower live weight. Okay, so in summary at this stage, our hypothetical question being, are there any harmful consequences of oversupplying copper? There seems to be quite a few benefits of oversupplying copper. We've got increased growth rates, we've got higher body condition scores, we've got an earlier first observed estrus, and that occurs at a lower live weight. So in theory, we may actually be undersupplying copper in the dairy industry at this stage. But our next slide tells a slightly different story. Now, the one criticism you could label here is that we started to AI these earlier. We did not. We started to AI at a fixed at 13.5 months of age. So any differences we have in fertility down here, and we have checked, are due to as a result of treatment as such. And if we look at pregnancy to first and second service, you can see that our conception rate for our control animals is all the way up at about 97%. In contrast, our animals on our high dietary copper concentration, it's down at 75%, and that's reflected in our serves per conception. Now, we've looked at this also as survival analysis, and we see no effect in survival analysis. These animals are eventually getting in calve. They're just being served very regularly to get in calve with a lower conception rate, but at the same quantity calve at the end as such. Now, what does this mean? It means that although we have animals that are growing faster, although we have animals with a greater body condition score, although they seem to come into stronger bulling, and indeed if you were to pull both sets of animals out, they would have looked obviously different, and you would have chosen the highs over the controls. But we do see potential implications on fertility as a result in this instance. Why this is occurring, difficult to know. I would say, given the capacity of the liver to keep the animal in copper homeostasis, we have some interaction going on with the liver there in terms of the hormones that are being generated that interact with the rest of our animal, whether it be something like insulin growth like factor 1, etc., something related to immunity and tissue mobilization, all of the above. Okay. So what was really going on in our liver at this point in time? So again, we biopsied them at 7 months of age, 13.5 months of age, pre-calving and then post-calving. Now, notice our dashed line in here is our limit for copper toxicity of 508 milligrams per kilogram dry matter. First thing to note is that obviously, our highs are higher than our controls, significantly different the whole way through, and that continues on to post-calving, roughly about 155 to 165 milligrams per kilogram dry matter difference. But the real sort of surprising thing here, and it came as a surprise to us at the time, is you see just how high both treatments are at the start at that 7 months of age. And we really think that that's a carryover effect. We have two things going on here. We have high copper concentrations in milk replacer. We also have high copper concentrations in our concentrates that we're feeding to our calves. That causes this mass increase in copper, liver copper levels at that point in time. This may also be compounded by the fact that that animal is transitioning over from being a pre-ruminant to a ruminant. Okay, during the pre-ruminant phase, sulfur and molybdenum don't combine within the rumen to form thiamolybdates, obviously. So that copper, that increased quantity of copper, is so much more available. So as an industry, we are probably vastly overloading the copper within our calves. Now, what consequences that has in terms of immunity, in terms of fertility, in terms of lifetime performance, it's difficult to know, because in this instance, we've actually done that to both treatments. But you can see that they subsequently decreased over into 13.5 months of age, where they remained relatively stable. So those differences that we got in fertility were actually within the normal region within the liver. So we may have implications for performance even within what we would consider to be normal copper concentrations within the liver. Now, not going to say very much about the post-calving phase. We did find a difference in milk yield in the first few weeks, where those animals on the control diet did provide a higher milk yield. Has that an implication for ketosis risk? It's difficult to know. These were first lactation animals. But probably the thing to highlight is that, again, we have this vast increase in copper in our left side, but moving from pre-calving onto our lactating diet, we get this vast increase in the copper concentration across both treatments again. And we have done another study to look at this, and I haven't presented the results here because it would be too big an animal. But the story to take from this is that that basal diet we're feeding and its effects upon things like rumen pH and all those interactions may have a much higher implication upon copper absorption and liver copper concentrations than we currently take account of. Okay, so in this case, it may be the transition from essentially a growing diet onto a diet containing high quantities of starch to drive milk yield. We subsequently get a reduction in rumen pH, copper becomes more available, and as a result, copper concentrations on both treatments increase, regardless of the quantity of copper being supplied in this instance. So what are our take-home messages? Overfeeding copper present on most dairy farms, and this is often from a whole variety of sources. Often nobody knows how much is being fed, and the problem is particularly acute with pre-ruminant calves. The high quantities of copper being compounded by the fact that the calf is a pre-ruminant. The animals look good, they grow well, they are in good condition and have strong bulling, but we may subsequently have negative consequences upon fertility. And I suppose the messages would be: analyze your forages and concentrates for minerals, take all sources into account, make one person responsible. If you see deficiency and toxicity, investigate it. There is no hard and fast rule when it comes to being conclusive about what has happened. And I would relate back to supple in terms of saying, simply try to find any instances of deficiency and toxicity, change supplementation as a result. And if you see altered performance, you can be quite conclusive that there was maybe something there. Thank you, Chair. That's me.
Thanks, James, for that very interesting presentation. There's loads of questions coming through, so keep them coming through the question box, and we'll come back to you after the rest of the presentation, James. So on to our next speaker, and I'm delighted to invite Professor Liam Sinclair from Harper Adams University to give his presentation around: can you feed low protein diets based on high protein forages without affecting performance?
Okay, Sean, can I confirm whether you can see my screen yet or not?
No, at the moment we can see lovely pictures of some cows on your desktop.
Okay. Has that done anything?
We can't see anything from your screen now.
You can't see anything?
No.
I'm struggling to see here just now.
Do you want to stop sharing, and when it comes through again, there might be an option to share your PowerPoint?
Okay. Has that stopped sharing? No?
Yeah. We're back to, oh, there we go. We can see Have you got two screens, Liam? We can see the PowerPoint now. You just need to put that in full screen, please.
Okay, you can see it now.
Yeah,
Yeah.
perfect.
Sorry about that, because it was on the other screen. Okay, sorry about the technical aspects. Thank you very much. My name is Liam Sinclair, and I'm going to be speaking about low protein diets for dairy cows based on high protein forages. So this presentation is really just taking in two themes of research that we've been working on for a number of years, both low protein diets and also how we can maximize the use of high protein forages. So I'm going to give a little bit of a background and then say a little bit about protein metabolism in the cow before we move on to some of the results that we found. So just as a general background, we are conscious in terms of feeding low protein diets because of the volatility of purchased protein feeds. And we can see here that we've got a large volatility in terms of the price of soybean meal, and other protein feeds tend to track these values as well. There's also a move away from certain protein feeds such as soybean meal, partly because of GM, but also because of what's called land use change, and if that soy has been grown in areas that have been deforested, particularly in terms of rainforests. So both of those are pressures to reduce protein and protein products. But we've also got environmental pressures as well. So the first would be aspects of nitrate vulnerable zones, which we're conscious of for a number of years across quite a range of the country. But more recently, we've had the clean air strategy. And the clean air strategy is focusing particularly on ammonia, and agriculture is the main producer of ammonia. And within agriculture, dairy cattle is the issue. And the problem with ammonia is that it combines with acids in the atmosphere and produces fine particulate material, what we call PM2.5. And that fine particulate material causes respiratory problems. So it's very similar to the issues that we've had with diesel cars, but we're focusing on the agricultural industry here. And as a consequence of that, there's been quite a bit of a pressure and incentive to reduce ammonia emissions from the farming community, and particularly dairy farming. But if we reduce protein levels too far, then there's a lot of evidence to say that that can reduce intake, and that can subsequently reduce milk yield. And that means that we can very soon lose the benefit from the savings and costs of feeding less protein, and we may actually start to increase our ammonia release per litre of milk if we reduce milk yield too far. So the other stream of research that we've been looking at is the use of homegrown legume silages. And they're attractive for a number of reasons. Because they are legumes, then they fix nitrogen, which means on a whole farm basis, we have to import less nitrogen onto the farm. So whole farm audits, it's got a benefit. But perhaps the main factor that we're interested in is that they are higher in protein than our traditional forages, such as grass and maize silage. So anywhere between 18 to 22% protein in the ration. Now, there's a number of forages that we can use as legume silages, but the two that are perhaps of most interest are lucerne, what the Americans would call alfalfa. And its characteristics are that it has a high amount of indigestible fiber, and as a consequence, its ME tends to be a little bit lower. But the fiber that is available is digested rapidly, which means the cows will end up eating more. Perhaps one of the major benefits is that it has a large taproot, and that large taproot makes it quite resistant to drought. So it is good in terms of drought conditions. The second one is red clover. Red clover is secondary to lucerne on a world basis. Lucerne is the most widely used legume on a world basis, particularly in North America and large parts of Europe. But red clover is perhaps more popular in many areas of the UK because it's more suited to higher rainfall, to cooler climates. And it also has a presence of this enzyme called PPO, polyphenol oxidase, and that can alter the release of protein in the rumen. because that's one of the problems that we have with these forage legumes, is that compared with what we're trying to replace, so soybean meal, rapeseed meal, distillers grains, etc., then the protein in these forage legumes is high in what we call rumen degradable protein. So it's very available within the rumen. And that can cause a little bit of an issue in terms of trying to balance the ration to avoid high levels of protein excretion. So I'm just going to take a minute or two and just explain a little bit about what's happening within the cow before we move on to our looking at some of our results. So if we consider the cow here, this is a cow giving about 32 litres of milk, eating about 4 kilograms of protein per day, so 4,000 grams. She's eating about 22 kilograms in total. That's what's coming in. In terms of what's coming out, then only about 25% of that is coming out in the milk. So 75% is being excreted in some form. So 4 kilograms coming in, 1 kilogram coming out. That can partly be related to body tissue changes, either mobilizing or depositing, but that tends to balance itself out. The main factors are related to the fecal output and the urinary output. Now, there's not a lot we can do about the fecal output because it doesn't vary tremendously, but there is with the urinary output. Most of the nitrogen that's coming out in the urine is coming out in the form of urea. About 80% is coming out in urea. So when that urea comes in contact with fecal material in passageways and walkways, etc., then that gets released into ammonia. So the urea in the urine is actually quite stable and quite safe. It's when it reacts with fecal material, it gets released into ammonia. So how do we reduce this urinary nitrogen output? Well, we need to consider a little bit more within the cow. And if we consider a little bit more within the cow, so this is our 4,000 grams of protein, and we call it protein, but what we actually analyze for is nitrogen, and then we convert that protein into nitrogen by multiplying it by a fixed figure. So what we're really interested in here is terms of nitrogen. So the protein or the nitrogen that comes in, most of it gets broken down by the rumen microbes. So this is the rumen here, and the rumen microbes breaks it down, and they break it down into ammonia. And these rumen microbes then use that ammonia to grow, which they then use when they're breaking down fiber and starch and other feed components. So they're growing within the rumen, and they're also getting washed out of the rumen when they're attached to feed particles or in the rumen liquid. So that's going to the small intestine. Part of the protein, however, in the diet, and that varies depending on the dietary ingredient, part of the protein bypasses the rumen, what we call undegradable protein, or it bypasses the protein, bypasses the rumen. And if we add this bypass protein along with our microbial protein, then we end up with what we would call metabolizable protein. And this is what we're actually interested in here, this metabolizable protein, because it's very similar to metabolizable energy. We don't consider rationing for cows based on the gross energy, we consider metabolizable energy. And equally, we shouldn't really consider the cow's requirements in terms of crude protein, we should consider it in terms of metabolizable protein. So if we feed a diet that's got too much rumen degradable protein, and forage legumes tend to be very high in rumen degradable protein, then what happens is that we end up with too much ammonia within the rumen. If we have too much ammonia within the rumen, it gets absorbed, goes to the liver, and the cow will then excrete that in urine coming out. So it is excreted as urea going out, and that can then contribute towards our problems. So we want to ensure that the rumen microbes have sufficient ammonia, but we want to avoid having excess, because excess is going to create that excess of nitrogen coming out in urine. And that's the challenge that we have in this project, is that we want to feed low protein diets, but we also want to feed them that they are including high levels of legumes that are high in rumen degradable protein. So the objectives of our study were to look at how we can feed low protein diets, but using these high protein forage legumes on performance, on nitrogen use, and on purchase feed costs. And to do that, we undertook 4 studies. The first was looking at reducing protein in red clover silage, then a lucerne, and then the third study, we tried to attempt to improve that microbial growth by adding starch and specific amino acids. And the fourth one is what we call a systematic review and meta-analysis. These are 3 individual studies. To really get a full picture, you need to look at all the studies that have been published in the literature. And that's what we did in this aspect here to give us a more meaningful picture. But I'm not going to say too much about that in this actual presentation. I'm going to focus on these 3. So I'm going to go through each one of the studies relatively briefly, just to give you a summary and some of the results. So this is the red clover study. So we had 3 protein levels, 17.5, 16.5, and 15. Importantly, they all had a similar metabolizable protein supply. So although they have different protein levels, they've all got a similar metabolizable protein supply. This one was slightly less than the others. With a ratio of red clover to grass silage of 1:1 or 50:50. And we grew the red clover as a single stand. We didn't grow it with the grass, and we did that. Most farmers would grow red clover along with grass, but we did that because the proportion of red clover in the sward can vary. It tends to be less in early season because red clover needs higher temperatures to start to grow, and it would be higher later. So to avoid that sort of confounding effect of not knowing what ratio we had, we grew the red clover separate, and then we added it in the mixer wagon with the grass silage. So a reduction in crude protein levels. In the Lucerne study, we had a similar approach. We had a 17.5%, so quite a standard level. We then went down to 15, which is more similar to this particular diet here. So these 2 are quite similar. And then on this one here, we went with the same low protein level, but we thought, well, let's try and use even more of the Lucerne in there and reduce the purchase feed even further by incorporating more Lucerne compared with maize silage. So in the Lucerne studies, we mixed the Lucerne with maize silage because farmers who tend to grow Lucerne are in areas where maize is also suitable. So this is more suitable in terms of the red clover, in terms of grass silage areas, and Lucerne in terms of maize silage areas. And we undertook a whole variety of different measurements, intakes, milk yields, milk composition, and I'm just going to summarize those in the next few slides. So this is our first red clover study. So this is our 17.5, 16.5, and 15% protein. This is dry matter intake here. And we can see that there's no effect in these 2, but it was reduced by about 1 kilogram in that one. So we had a lower dry matter intake. On the Lucerne study, so this is the 17.5%, 15% protein, and then 15% protein with more Lucerne. Then again, we had a reduction in intake, but of about 1 to 2 kilograms of dry matter. However, despite that, we had no effect on milk yield. So this is the milk yield in the red clover study, which was similar across all 3 treatments. And in the Lucerne study, it was also similar, but when we pushed the Lucerne level of inclusion that little bit higher, then it came down a bit. So a bit of a reduction when we went to even higher levels of Lucerne inclusion. Milk fat and protein, no effect of protein level on fat and protein in the red clover study. And similarly, no effect on fat and protein in the Lucerne study, although again, we had this effect here on the low protein diet with the highest inclusion of Lucerne, that it was slightly lower. Moving on to look at aspects of milk urea, of nitrogen use efficiency. So the first is milk urea, which you'll be familiar with in terms of bulk tank figures. And there's a very strong relationship between milk urea and how much is going to be excreted in the urine. Generally, you'd be after values, you don't want to be 25 to 30. So even our high crude protein diet was quite low here. But nevertheless, we had this decrease in milk urea as we reduced the protein level in the red clover study. And similarly, we had a reduction here in the Lucerne study when we reduced the protein level, irrespective of how much Lucerne we included. So there's lower milk urea and therefore less nitrogen being excreted. And when we look at the nitrogen use efficiency, so this is how much nitrogen is being captured as a proportion of that being consumed. So when I showed you that earlier slide with the picture of the cow, we said it was about 25, and it was about 25 in the high protein clover. And when we increased it, particularly when we got down to 15%, we were up at over 30, 31% nearly in terms of nitrogen use efficiency. So quite a large increase going down to that lower level. Similarly, on the CERN study, nitrogen use efficiency was increased when we reduced the protein levels about 26%, going up to about 32%, but nevertheless, there was an improvement. So those are our first two studies, but what we didn't have a very good handle on in those first two studies was aspects, longer term aspects in terms of live weight and body condition. So our third study was focusing on red clover, because there's much less information in the literature on red clover. Most of it is on the CERN. So we decided to focus on red clover. Again, we had a 17.5% and a 15% protein diet. And then with two other low protein diets, one where we added additional starch, and that was to provide more energy for the rumen microbes to grow. And if they grow, then that's going to do two things. Firstly, they're going to capture more ammonia within the rumen, so less available to be excreted. And secondly, they are going to grow themselves, and that's going to supply more metabolizable protein. The fourth was to look at the quality of the metabolizable protein. So what the cow actually uses is amino acids, and the amino acid that should be short of most is methionine. So we added rumen-protected methionine in this treatment, still 15%, but rumen-protected methionine to try and improve the quality and therefore improve animal performance. We used 56 cows in this. We took them over 14 weeks, and again, we did a number of measurements. Again, the important point here is that the metabolizable protein supply is similar, although the protein is varying. They're slightly deficient in these three diets so that we could detect the difference a little bit easier, but nevertheless, the metabolizable protein was varying relatively little, whereas the crude protein was a lot. In terms of intake and milk yield, then we had no effect on intake, unlike the previous studies. There are differences in individual weeks, but no effect on intake and no effect on milk yield. So we can see that they had a similar milk yields varying a little bit over a 14-week study, but there was no effect over that period of time. Similarly, no effect on milk fat and no effect on milk protein. We did find that cows that were given the additional starch tended to be, had a higher body condition score than those that didn't receive the additional starch. And that's not unusual when you feed more starch to improve body condition score between the different treatments. In terms of milk urea, then similar to our previous studies, when we reduced the protein, we had a lower level, so we had less milk urea, and we did actually measure the nitrogen output in these cows, the urinary nitrogen output, which I'll mention in a minute. And our nitrogen use efficiency was a bit higher in the control one here at 28, but it went up to about 34–35% in our treatment diet. So again, we had quite a large increase in terms of use efficiency. So what about the feed costs? Well, I've looked at the concentrate costs here, and this is across the 4 treatments in the last study, and I've expressed it as pounds per cow per day, per kilogram of milk, or per kilogram fat and protein. And there is a saving when you reduce the protein levels because you're feeding less purchased protein feed. However, not perhaps as large as you would expect. And when you express it per litre of milk, the low protein with starch was the best. It was about 7 pence, a saving of about 0.7 pence a litre, whereas the protected methionine, because it's quite expensive, was similar. There wasn't much saving in costs. And overall, the saving in costs, so we had a similar intake, a similar milk performance, but not perhaps as large a saving in costs as we would expect. And the reason is that we had to feed more expensive protected protein sources in these lower protein diets. So to maintain that metabolizable protein supply, we had to feed more protected sources in there. So protected soya, protected rapeseed meal to provide that, maintain that protein supply. So we didn't have as much savings as we would expect, but we could have a saving in this study somewhere between 0.5 and 1 pence a litre. So the take-home messages are that we can reduce dietary protein level to about 15% or 150 grams per kilogram dry matter in these high protein forage legume diets without affecting intake and milk performance. But the most important factor is that we maintain the metabolizable protein supply. So that is the critical factor, not crude protein. We really have to move away from crude protein. The hint is in the name. It is crude, and it doesn't really tell us very much more than what we need to know. We need to look at the metabolizable protein. We found that by reducing that, it increased the N captured in milk and reduced the amount excreted when we measured it in the cows by 66 grams of nitrogen per day. That may not mean very much in terms of what you may be looking at, but that's equivalent to 20 kilograms of nitrogen per cow per year, or about 50 kilograms nitrogen per hectare, going back onto the land. And remember, this is predominantly in the form of urea, so it may not actually get onto the land because it may be released as ammonia into the atmosphere. So if you take that into consideration, that's quite a large reduction. And we can make savings of up to 1 pence per litre from on-purchase feed costs, but we've got to be conscious that to get down to these lower protein levels, we've got to feed more expensive supplementary feeds. We feed less of them, but they are more expensive, and therefore our savings are perhaps not as large as we might have anticipated. Okay, and I shall finish at that.
Lovely, thank you very much, Liam, for that presentation, and we're running bang on time, so we will come back to questions after the next few speakers. So thank you very much for your presentation. Just a reminder for those that have joined us after the introduction that we do have a Q&A session at the end of all the presenters, and I would encourage you to type in your questions into the question box that you can find on the right-hand side of your screen. So we'll now move on to our third presenter today, and this is Professor Phil Garnsworthy from the University of Nottingham, and Phil will be presenting around what impacts your dairy farm efficiency. So over to you, Phil.
Can you see my screen?
We can see your desktop at the moment, but no presentation.
It's the wrong screen then. Show screen. Is that one better?
Yeah. Oh, yeah, perfect. There you go.
Right. Okay. So hopefully you can see my opening slide. So today I'm going to talk about what impacts your dairy feed efficiency. I'm just going to set up my pointer. Right. So first I want to thank my co-authors, particularly Emma Gregson, who was the postdoc that worked on this project, and she's the one that led all the on-farm studies, and Jen Goodman, who was a technician who went and helped Emma. Jean Margerson inputted some information on young stock, and Paul Wilson ran the farm business survey. Next slide. Okay, so this project was a work package on our AHDB research partnership with Nottingham University, and the idea was to look at whole farm feed efficiency, and one of the key things was to answer questions that farmers might have. So the first question might be, how efficient is my herd? How does it compare with other herds of a similar system? What can I do to improve efficiency, and is it going to be worth it? Will it cost me too much money to improve efficiency? So the methods that we used were, first of all, to define dairy systems, because different systems will have different sort of targets for efficiency. So an all grazing system would have totally different targets than a housed or year-round system. So we had to define systems. We did a on-farm survey where we visited farms throughout a whole year and took detailed measurements. We did a farm business survey where we took 300 farms at random from the National Farm Business Survey and asked extra questions so that we could calculate their feed efficiency. We did that for 2 years. We did some on-farm studies looking at feed use and feed wastage, which I'm not going to go into today. And then we put everything into a model so that we could look at what parameters farmers could concentrate on to try and improve their efficiency. So the outputs from the project were definitions of the systems, a calculator for calculating feed efficiency, which is going to be incorporated into EnviroBench, what drivers there are of whole farm feed efficiency, and what are the benchmark key performance indicators. So the first thing we have to do is to define whole farm feed efficiency. Now, people think of feed efficiency usually for individual cows or milking cows as the milk they produce divided by the feed that they eat. But we took it a step further and we looked at the milk produced on the farm, so the total milk divided by all the feed purchased and produced on the farm for all the dairy animals. So we include not just the milking cows, but also dry cows, replacements, and any feed wastage that there is. And we measured feed on a dry matter basis, and milk we express on an energy corrected milk yield basis. So our first task was to define dairy systems so that we could have different targets and different approaches for different systems. So we got together a whole group of people from data recording companies, from consultants, from advisors, from feed companies, and obviously with AHDB as well. And we went through all the different things on dairy farms that we could use to distinguish them easily. And the main factor that we came up with was the number of days that animals spent grazing. So we defined the first system as where cows spend more than 9 months of the year at grass. And then the second is between 6 and 9 months. System 3 is between 3 and 6 months. System 4 is between 0 and 3 months. And system 5 is housed all the year round. So that's a very simple system to for farms to say, how long do I graze my cows, and put them into the various systems. And that sort of maps onto calving patterns as well. So the ones that are grazing for more than 9 months of the year tend to be spring calvers. The ones that graze for 6 to 9 months may be autumn or spring blocks or all year round. And similarly with the ones that are 3 to 6 months. And then the ones that graze for just 3 months or they're housed tend to be all year round calving. So as I said, we did a survey. We visited 21 farms, and they were representative of all 5 systems. And Emma and Jen visited them quarterly between April 2017 and March 2018. And they did a huge number of recordings, looking at everything to do with the animals, the number of animals and when they calved and things like that, looking at milk production, milk composition, looking at feed use, all the feed grown on the farm, the forage and the grazing land use. They measured grass intake, but this was done by looking at the amount of grass in a paddock before cows went in and the amount afterwards, and saying that the difference was what cows had eaten. This is complicated because grass grows, but they adjusted for that as well. And they also did some studies looking at feed wastage on farms. So to cut a long story short, we put all these numbers into a calculator and looked at the feed efficiency for the different dairy production systems. So you can see that system 1, grazing for more than 9 months of the year, had an efficiency just below 1. And as we reduced the amount of time grazing, so efficiency increased, and the housed all year round had an efficiency of 1.13. Now, we're not suggesting that farmers should change from one system to another, or at least not dramatically. They might go from a system 2 to a system 3 or something like that, but they wouldn't have wholesale changes from grazing all year to housed all year, or vice versa. But the key thing is that we can look within all these systems, and we found a wide range of feed efficiency within systems. And overall, the lowest efficiency we got was 0.5, and the highest efficiency we got was 1.5. So within systems, there's quite a bit of scope to improve efficiency. Looking at the economics of feed efficiency from the Farm Business Survey, this was the first year's results. 194 farms gave complete information that we could calculate from, and we see that for every 0.1 increase in feed efficiency, we were getting £233 extra gross margin per hectare. And looking at the gross margin and net margin of the 5 systems, there was no effect on gross margin for most of the systems except the housed all year round gave a higher gross margin per hectare. And in terms of net margin, we found that the 9 months grazing or the housed all year round had higher profits or net margin than the other systems. Looking at forage types, remember this is about 300 farms. As the proportion of time spent grazing decreased, there was decreasing use of grass silage, increasing whole crop, and increasing maize silage. And also we got increasing use of concentrates up to the system 3, the 3 to 6 months, and then it was fairly steady thereafter. But certainly the ones that are focused on grazing use less concentrates. So what we found were the main components of whole farm feed efficiency. First of all, it was the number of cows on the farm and the milk yield per cow, as you might expect. The length of lactation, fertility and health, and longevity of the cows. In terms of heifers, then the first thing is the number of heifers on the farm, and that is determined by the replacement rate of the herd. It also is affected by age at first calving and health of calves and loss of heifers during the growing phase. For feed intake and wastage, then the main driver was the stocking density, whether that's cows at grass, the stocking rate there, or whether it's the number of hectares used to grow forage for housed all year round systems. So it depended what feeding system people had, the quality of the feed, how well they managed clamps, and whether feed was recycled to heifers. So this can give an increase in feed efficiency of the whole farm if some of the TMR that's left from the dairy cows is fed to heifers, but obviously you need to make sure that you're still giving the heifers a good diet that's well balanced to their requirements. And we considered all the different feeds, so graze grass, forages, straights and blends, concentrates, and calf milk. Now, looking at feed efficiency, then the first thing we can look at is milk yield. And this is some figures where we show the number of cows required to produce 1 million litres, which is the blue line, versus the energetic efficiency of those cows, which is the red line. So if you've got a herd yielding 4,000 litres of milk, then you need 250 cows to produce 1 million litres. If your cows yield 10,000 litres of milk, you only need 100 of them. And as you decrease the number of cows or increase milk yield, then energetic efficiency increases because higher milk yields dilute maintenance per cow. So the total energy required by the cow is more for milk and less for maintenance. And it dilutes it per 1 million litres because you need fewer cows to produce 1 million litres. But higher milk yields can sometimes be associated with inefficiency. So if you've got higher milk yields, then sometimes people might expect lower fertility, higher health risks, earlier culling, and a higher replacement rate. And the high replacement rate will affect lifetime yield. There'll be more heifers on the farm, so you're using more feed for non-productive animals. And the other heifer factors that play a part are what your target weight and age at calving are, the actual age at first calving, losses of heifers, and what the heifers are fed. And we were interested in how these things interact and how they affect whole farm feed efficiency. So the main drivers are milk yield per hectare, the stocking rate, grass and forage quality, and the proportion of feed used for replacements, which is affected by number of replacements, the number culled for fertility, the health of the animals, and the longevity. And these factors aren't independent. You might get no response, but some sort of interaction underneath. So looking at milk yield per hectare, this is Farm Business Survey data. You can see that as you increase milk yield per hectare, then you get an increase in efficiency. It's a curvilinear response, and milk yield per hectare is a key driver of whole farm feed efficiency. Stocking rate, this is from our survey. There was really no effect on efficiency once we got above one livestock unit per hectare, so that's a cow plus their replacements. But it was very low efficiency for these farms here, where they weren't really using grass as a major nutrient source. It was just to give cows access to fresh air. So stocking rate is a key driver when you've got low stocking rates. Looking at grass and forage quality, we have grass quality on the left, looking at metabolizable energy content of grass. And as you'd expect, the more reliance on grass, the higher the response. So system 1, 9 months grazing, at a very steep slope. So the higher the quality grass, the better the efficiency. And then it's system 2, system 3, 4, and system 5, there's no response because they weren't going out to grass. Looking on the right, we have grass silage quality, and we get the reverse of the system effect. So the highest slope is for system 5, where they're housed all year round, more reliant on silage, and the lowest slope is system 1, where they're only housed for about 3 months of the year. But better grass and forage quality will always increase efficiency. Looking at the non-feed factors, then these were some of the main things that we looked at. Mastitis rate didn't have a major effect on feed efficiency. And this is because cows that have mastitis tend to have it for just a week or so, and then they might get sold or they recover. So the reduction in milk output is quite small, and they tend to eat less food when they've got mastitis as well. So it had a very small effect on feed efficiency. Culls for fertility was the biggest factor we found that affected whole farm feed efficiency, and this is because the more animals you cull, then the higher the replacement rate, the more heifers on the farm. Age at first calving had quite a strong effect as well. The longer you keep your heifers before they calve, the more food they're eating without producing milk. Loss of heifers in their first year, again, it didn't have a great effect because they won't have eaten a high proportion of feed. Loss of heifers greater than 1 year of age does have a bigger effect. Fertility effects on efficiency, they're different for different targets. So in seasonal calving herds, we're interested in proportion conceiving by 90 days. We're aiming for, by 100 days, we're aiming for 90% or greater. And you can see if you have lower proportions, then efficiency decreases. For all year round calving herds, we're looking at conception rate and estrus detection because they multiply together. And if you've got a very good conception rate, 60%, then estrus detection doesn't have a great effect unless it's down at 50%. But if you're down around 30 to 40% conception rate, then estrus detection rate becomes increasingly important. And finally, to look at why we're talking about replacements so much, this is the energy required to produce 1 million litres of milk at different replacement rates. So the energy required by the cows is not going to change. You're producing 1 million litres of milk, but as replacement rate increases, then you get increasing amounts of feed required by the replacements. And the different colours for the replacements are different ages at first calving. So if you go from 24 to 30 to 36 months, then you're increasing the amount of feed required by those heifers. And looking at it overall, then over this range, feed energy required for the heifers range from 16 to 44% of the total feed energy required for the herd. So if you've got a really high replacement rate, a late age at first calving, then nearly half of your feed is going to heifers. So a high replacement rate or a late age at first calving reduces whole farm feed efficiency, it reduces margins, and it will increase methane and nitrogen outputs. So just to conclude, then how can we improve whole farm feed efficiency within systems? The main target is to improve milk output per hectare, and you can achieve this by improving grass and forage quality, by better control of grazing, better control of forage when you cut forage and clamp it and things like that. Better utilisation, so making sure that the good grass and forage is well balanced with the correct nutrients. And then you can increase stocking rate per cow or increase milk yield per cow at the same stocking rate. That will improve efficiency. You need to manage feed to reduce wastage, manage young stock to reach their age and target weights at calving, and maximise fertility and health to reduce wastage of animals. So thank you. Thank you for your attention.
Thank you, Phil, for that excellent presentation. We do have questions coming through, but in the interest of time, we will move on and come back to you. So thank you for the time being. And just a reminder to the audience, if you are popping a question in the chat box, please can you state which presenter your question is for, just to make sure that we get the right person to answer your question. Thank you very much. And moving on, I am delighted to welcome our last speaker of this morning's session to the virtual stage, and Brad Harrison is going to be discussing phosphorus management in Great Britain dairy farming. Where are we and what can we achieve? So over to you, Brad.
Thanks, Cyan. Can you see my presentation?
Yeah, you might just want to pop that in full screen and then we're good to go.
There we go. Is that it?
Yeah, perfect.
Awesome. Hi, everyone. It's a tough group of panelists to follow. So it's a very star-studded cast, but AHDB basically asked me to give a quick 5-minute presentation on one of the papers that came out of my AHDB funded PhD that I finished earlier this year. And it was all around phosphorus management in GB dairy farming systems, but more in particular, it was around phosphorus balances across the systems. So I can't go into as much detail as the previous presentation, so consider this a bit more of a higher level teaser of the paper. But the very first question is, why do we care about phosphorus management? So improving phosphorus use efficiency has a long list of benefits. So at the farm scale, we can reduce eutrophication. From the farm scale, we can reduce eutrophication in surrounding water bodies. We can increase net profits by reducing the amount of imports. At the national scale, we can improve national food security by reducing our reliance on imports from other countries. And then on the global scale, we can slow the depletion of finite phosphorus reserves. So where are we in terms of phosphorus management? So I've included some loose figures in the results on dairy farming phosphorus balances, but essentially it's a very difficult question to answer. So in countries that have diverse dairy farming systems, such as Great Britain, we have limited information on phosphorus balances. And although information is available, they're becoming less representative of modern GB dairy farming as it moves to more diverse systems. Fewer of that information uses actual measured phosphorus concentrations of the imports and exports into and out of the farm gate of a dairy farm. And then even fewer actually compare these phosphorus balances between the dairy farming systems. And then there's almost none of the information is available for soil phosphorus balances, which is the phosphorus imports and exports into and out of the soil surface. So what were we aiming to do around this issue then? So the main aim were to determine the phosphorus balance and use efficiency and identify the key determinants of phosphorus balance and use efficiency across a range of dairy farming systems that operate in Great Britain. So today, the results I'll be showing is more related to the determining the phosphorus balances as opposed to the key determinants for the sake of time. But the implications of this would be that we could contribute information towards developing strategies to improve the sustainability of dairy production in these countries that operate more diverse dairy farming systems. So how are we going to achieve this? So what we had was we selected 30 dairy farms from a pool of volunteers, and we selected the farms to try and represent each of the 5 GB dairy classifications that, surprisingly, Garnsworthy actually explained in his previous presentation, which is based on the calving approach and the day's access to pasture. Each of these farms that we selected then completed a phosphorus balance report that we specifically designed for this project. And then each of the farms were visited once between, I think it was October to March. And we collected samples of their feed, manure, and soil to analyze for phosphorus concentrations. And then using all this information we gathered, we run the data through the principles of the planet model to calculate farm gate phosphorus balance. And then we calculate a soil phosphorus balance using the principles of the anchor. So that's the annual nutrient cycling assessment tool from Wageningen in the Netherlands. And essentially what that all gave us was, so what's presented here is 2 data sets. So firstly, the 9.6 here is the mean farm gate phosphorus balance of the farms in the current study. I've added some benchmarks of previous, well, data of previous larger scale studies from around 2005, 2006. So the farm gate phosphorus balances were about 15.3 to 20.1 kilograms of phosphorus. And then for the soil, the mean soil phosphorus balance for the dairy farms in the project was 7.5 kilograms of phosphorus, which was similarly a bit lower than what previous larger scale studies have found. And there's a whole wealth of limitations in comparing this study with these previous studies. But what is relatively safe to say is that where we took our farms from a volunteer pool, it's likely that it's representative of farms that are more interested in phosphorus management than perhaps the average farmer. The second set of data amongst this table, as you can see, the 5 classifications are separated out here. So the letters, the A's and the B's, are relevant to the statistical test we used, which was ANOVA, and essentially classifications that don't share the same letter were significantly different. So what we can see here is the blended dairy farming systems. So classification 2 and 3 were actually had significantly lower phosphorus balances than the housed system. So for a quick roundup then, in terms of where are we with phosphorus management, the study doesn't necessarily contribute the most representative data towards that question, but what it does do is promote the angle that where we are with phosphorus management is probably dependent on the dairy farming systems that are being used. What we can achieve is similarly dependent on the system that we're using. And how can we achieve this was explained a bit more in the paper, but essentially it was system-specific practices such as the house systems feeding more closely to phosphorus feeding recommendations, importing less phosphorus of concentrates, and then some pasture-based systems could do increase the inclusion rate of homegrown forages, particularly forages that are homegrown, and matching the stocking rate with the availability of forages. And then all systems could benefit from more accurately crediting the phosphorus in soil and manure. But also a final conclusion that we got from the project is that we used the foundations of a novel approach that could be developed further to create a system where we can calculate phosphorus balance more easily and robustly for farmers that operate a more diverse dairy farming systems. So just to finish, I just wanted to thank the AHDB for funding the project, all my co-authors, the staff at Wageningen University, and all of you guys, so the advisors, farmers and societies that made it possible. And then I've got the slide for the full paper at the end there.
Lovely. Thank you very much, Brad, for that quick run through. So people will be able to capture the details you put in your papers, and would encourage you, if you want more information about the teaser that Brad's just provided you, to visit those papers. And the final report for Brad's project will be available on the AHDB Dairy website in due course as well. So again, a big thank you to all our speakers this morning for all of their excellent presentations. I hope you found it very interesting. I would like now to invite all our presenters to join me on screen, and we will kick off the Q&A answer session. And for this, I'm also going to invite my colleague Doreen to ask your questions. Thank you.
Yeah, good afternoon, and a huge thank you to everybody. There is loads. I'm scribbling away quite frantically here, trying to make sure that I've got them all written down. So I'll try and keep them in some sort of order. James, can I come to you first? The first question was, you mentioned copper is released back into the blood. What is the mechanism for this storage, and what controls the release?
Really, you're under homeostatic control. ATP7B, ATP7B translocate, and then you get the synthesis of cereoplasmin and its release in plasma copper into the bloodstream as a result, and feedback according to what levels you've got there.
Okay, another one for you then. So somebody has written in, as far as I'm aware, our organic diet contains no added copper. Would our soil and silage analysis give us enough info to estimate whether our cattle are getting what they need?
They should do. I mean, you're going to work off essentially historic concentrations. If you're getting feeds analysed, concentrate should be relatively bog standard analysed. You take that value. If you then got the analysis of the forage that you're feeding, you can then work out relating to requirements, and most dietary software will give you an indicator of where you are in relation to that requirement anyway. And some of them will take into account sulfur and molybdenum concentrations as well. But again, it's taking that with a word of warning, because obviously you may have dietary factors in there that will not be taken into consideration, but have effects upon copper absorption and the animal.
Okay, super. I'm going to stay with you, James. I'm sorry, there's quite a few here for you. Did you look at the genetic merit of the heifers at all?
Yes. So they were blocked according to predicted transmitting ability, which was the best that we could do, because you don't have 305-day milk yields, obviously. So they were blocked according to predicted transmitting ability for milk yields, fats and proteins. And then you also have in there live weight, which would give you an indication of growth rate and body condition score and wether height as well. I think we had, and somatic cell count.
Okay, thank you. And is the absorption of copper in calves higher than in adult animals?
It will be. I mean, the bit that I neglected to mention is that as humans, we're actually very different. I mean, we don't get up in the morning and say, I need to take my copper tablets today. And the reason being is that we're essentially developed to absorb vast quantities of copper as monogastrics and pump it all back, or excrete the excess back out. Ruminants are quite contrasting in that they're designed to absorb. So that absorption coefficient as a calf transfers over from being a pre-ruminant to a ruminant animal will probably you're talking about probably 70% absorption potentially in the calf, down to 0.07 absorption as an estimate as a ruminant animal, as a fully functioning ruminant.
Okay, I'm going to ask you one more, and if I've got time, I'll come back to you. Accepting that sulphur impacts on copper availability, did you look at sulphur levels in forage diets for young stock, bearing in mind the increased use of sulphur in fertilisers on grassland to compensate for reduced atmospheric depositions?
I'll just say that one again for me. I'm trying to track through.
Accepting that sulphur impacts on copper availability, did you look at the sulphur levels in forage diets for young stock, bearing in mind the increased use of sulphur in fertilisers on grassland?
I mean, we did record. Obviously, we recorded our sulphur concentrations the entire way through for all the different diets that they would have been fed, and combined them to get an average overall. We found our sulphur concentrations to be, from memory off the top of my head, slightly higher than we would observe in other farms. So those results are within that frame, that our sulphur concentrations were slightly higher. Would that have had a serious effect? Probably not. I think the main thing to consider when it comes to sulphur and molybdenum concentrations are exceptionally variable within farms. And is that as a result of practice? I don't know. But I think the best indicator of your sulphur and molybdenum when it comes to what you're about to feed is probably that sample at the clamp, which gives you an idea of your overall sulphur and molybdenum concentration.
Thank you. Right, I'm going to come back. I'm going to come to Liam. How would you suggest using this knowledge in an organic system? Average yield around 9,000 litres in mid-Cornwall, whether that makes a difference.
Yeah, I think the results are very applicable, because organic farms often do tend to feed quite high amounts of forage legumes in there. So I think the results are particularly applicable to those. And the basic message is that you can feed lower protein levels, but you need to make sure that the metabolizable protein supply is correct. Now that might be a little bit more challenging in terms of getting that bypass protein through that I spoke about, that you needed to feed higher levels of in terms of what sources you're allowed to use. But the principles are essentially the same. If you want to reduce the excess rumen degradable protein, you've got to reduce your protein levels and make sure that you're supplying that metabolizable protein supply. So it's moving away from this focus on crude protein, but I think the messages are that we can do that with high yielding cows and do it with forage legumes. So the results are very pertinent, I think, to organic farm.
The second question is there any data on fertility effects with the different CP levels, particularly in your study 3?
Well, our study wasn't long enough to do that, but they have completed another study at Reading University, where they have taken animals 200 plus heifers through from calves through to their third or fourth lactation. So Chris Reynolds was involved with that. So they were looking at 18%, about 16% and 14%. And they found that if you fed very low levels of protein, 14%, that they tended to have more issues with fertility and with aspects of some aspects of lameness, but it didn't appear until about the third lactation. Now, the important point to remember there is that although they were feeding 3 different levels, they didn't keep the metabolizable protein the same. So perhaps if they'd kept the metabolizable protein level the same, the differences may not have been there. Other studies from the literature would indicate that when you reduce protein, that you don't have a negative effect in terms of fertility or lameness or other aspects of health. So again, it comes back to the point that we should be focusing on metabolizable protein rather than what the actual crude protein content of the diet is. But if you do get those wrong and you do supply insufficient, then the evidence is that it can have a negative effect, but it's a long-term negative effect. So even in our study, I don't think we'd have picked it up because it wasn't long enough. And the work at Reading, it took 3 or 4 lactations before they actually started to get differences coming through, but they were under supplying metabolizable protein. We weren't. We were supplying the cow's requirements.
Okay, last one. Are there any benefits of feeding lupin silage as a source of metabolism protein?
Yeah, I think lupins are, they're another source as well. I think, growing homegrown protein sources does reduce the, the volatility of world markets in terms of buying purchase feed. But of course, they do expose you more to the volatilities of climate because you're growing it on farm. So, it kind of swings and roundabouts in some ways in terms of when you lose and when you don't. But lupins, if you can grow them well, then yes, they can be included in. Again, you've got to have the diet balanced properly to take into account their supply of ruminant-gradable protein and undegradable protein, the same as you would with any other forage.
Lovely. Okay, Phil, I'm going to come to you for one big question probably. High yielding is more efficient, but will they reduce fertility, health and increased culling rates cancel out this efficiency? And how would a farmer work this out?
Yeah, and that was the main point that I was making at the beginning, is that in theory and generally in practice, the higher the milk yield, the greater the efficiency. Certainly the higher the efficiency of the cows, but if you have too high a replacement rate, then that will cancel it out. So it could be poorer fertility, more health problems, but basically sort of you've got a lot more heifers on the farm than you need. So farmers should be able to work out the replacement rate fairly easily. They just count the number of cows they've got in the herd and how many they cull each year or how many heifers they bring in each year. In a balanced herd, then the number of culls should be the same as the number of heifers coming in. So that's your replacement rate. So it's fairly easy to calculate. In terms of calculate how much feed the heifers get, then that's much more difficult, because often heifers are put in the furthest field away from the farm, or they're put in a barn and fed on poorer quality silage and things like that. But it's up to sort of individual farmers to check the growth of their heifers and make sure that they're growing well.
Lovely, thank you. There was one question that came in. Is the Dairy Point Pro points for this? Yes, there is. If you can put your details into the questions and we'll pick that up as well, so we'll add it to your Dairy Pro. And the final question, I'm conscious of time, is for Brad. Can you explain the difference between planet farm gate balance and soil balance?
Yeah, sure. So the planet farm gate phosphorus balance is at the farm gate scale. So it's imports such as feed and mineral fertilizers, and then the exports, which is largely to do with milk. And then what the anchor tool does is concentrate on, well, we took the aspects of the soil surface phosphorus balance out, which looks a bit more into the imports into the soil surface and out of the soil surface as crops. So the planet farm gate version is looking a bit more at the efficiency of feed, because that's a large amount that comes into the farm, whereas the soil surface phosphorus balances are looking a bit more about fertilizers like manure, mineral fertilizers, and the productivity that comes out as crop.
Okay, lovely. Thank you, Siobhan. I think I've captured everything. If there's anything else, we can always go back individually at the end. So I'll pass it back to you.
Lovely, thank you very much, Doreen, and thank you to all our audience for submitting their questions. And again, a big thank you to all our presenters today for their presentations and for their answers during this Q&A session. We are coming to the end of our webinar now. I've just got a few points to run through with you before we finished with a final presenter that is coming to you pre-recorded today. So just a note again to please complete the feedback survey that will appear on your screen once this webinar ends. It really does help us just make sure that we are delivering these around topics and to the standard, to a high standard. The video will be sent and the recording within 24 hours, and the slides will also be made available online, so do keep a lookout for those as well. So thank you very much to everyone for joining us and giving their time to join the webinar today. And as I mentioned, to finish this session, I'm delighted to introduce a pre-recorded video from Chris Hudson on behalf of Ed Pace, a PhD researcher at the University of Nottingham. And the video will cover evidence synthesis to improve dairy herd fertility.
Hi there, I'm Chris Hudson. I'm a clinical associate professor here at the University of Nottingham Vet School, and I'm down on our dairy unit today to give you a bit of background to an AHDB funded project that I've led over the last few years. So the background to this work came out of experience really of working with dairy farmers to improve fertility performance, and the knowledge that fertility is actually an area where we have a lot of understanding from research about the different factors that can affect it. So in contrast maybe to something like lameness, where there are quite a few aspects of disease that we actually just don't understand that well, fertility is very well researched, and there's a very, very big body of quite robust and high quality research out there. The problem I've often found is that it's quite difficult to put that into practice. And it's particularly difficult because studies tend to look at one specific aspect. Studies use different measures of reproductive performance, and it's really difficult to know, given that, the knowledge that a cow that's had subclinical ketosis, so kind of extreme negative energy balance in early lactation, her chance of getting pregnant to a particular serve is reduced by X% in the first 100 days in milk. It's very difficult to use that for a farmer. So it's very useful to know, but actually it's quite difficult to know how much emphasis should we be putting on improving early lactation energy balance, and how would that compare to investments in other areas. And alongside that, I was also aware that the relationships between post-calving disease and reproduction are actually not as well understood as I'd thought they were. So the two outcomes that we were looking to achieve from this project were to improve, update and refine our knowledge about the relationship between diseases like retained fetal membranes or metritis and reproduction, and to look at how those relationships might change at different levels of milk yield or different parities of cow, and how do they change as a cow moves on through lactation. And alongside that, to put the results of that work into a simulation model and to build in other research knowledge that comes from other people's published research outputs across a variety of other different areas that might affect fertility. So we'll have our post-calving disease results in there, but the simulation will also include results from other people's work on different aspects like heat stress and infectious disease, and measures that you can implement to change heat detection or submission rate. So there's a couple of things we're aiming to do with this simulation model. One is to use it to look at what you would expect to change under various different common scenarios. So we could say, okay, for this broad type of herd, these are likely to be the interventions that are actually most effective in most situations. And even more excitingly, to develop a decision support tool, which would be an online tool whereby farmers and their advisors could enter their starting situations. So they could say, this is our current level of reproductive performance, this is our infectious disease status, this is the proportion of cows that get retained fetal membranes or metritis, this is the proportion of cows that have subclinical ketosis in early lactation, and so on. And then they could look at, okay, well, if we change this particular aspect, if we adopt this particular approach to heat detection, or if we reduce the rate of post-calving disease, what would we expect to happen to our fertility performance, and how confident does the research allow us to be about that predicted change? So it potentially is a really powerful way for producers and their advisors to compare different interventions that they can make to improve fertility performance. And I guess most importantly for me, it puts that huge body of research knowledge right at producers' fingertips.